Quantitative estimation of protein using BIURET reagent, multi-standard calculation as well as graph is used. The reagents involved are BIURET reagent, 0.1 normal NaOH, working standard of 200 milligram protein per 100 ml, two unknown concentrations of test 1 and test 2, 7 test tubes marked blank, S1 to S5 serial dilution of standards, then T1, T2, two unknown concentrations of protein solutions. We need glass pipettes, teats, and if you want to use micro pipettes can be used. We need a water bath of 37 degrees Celsius, a colorimeter or a spectrophotometer if you have one. We start by adding different amounts of working standard 200 milligram per 100 ml of protein to S1, S2, S3, S4 and S5 in the order 0.2, 0.4, 0.6, 0.8 and 1 ml. Now this is followed by addition of the unknown concentrations test 1 and test 2. 1 ml each of test 1 is added to tube T1 and test 2 is added to tube T2. All the test tubes are made up to 2 ml using 0.1 normal NaOH. First of all, the blank test tube we add 2 ml of NaOH, then from S1 to S5 we add 1.8, 1.6, 1.4, 1.2 and 1 ml of 0.1 normal NaOH, and to the tubes marked T1 and T2 we add 1 ml each of the this particular solution, that is 0.1 normal NaOH, so that all the tubes will have exactly 2 ml of solutions in them. This is made up to 2 ml, all the test tubes, including the serial dilution of standards. Next, we add BIURET reagent, 3 ml to each of these test tubes. 3 ml of BIURET reagent is added to blank from S1 to S5, T1 and T2, so that all the test tubes will have a total volume of 5 ml. The tubes are now incubated at 37 degrees Celsius for 10 minutes, and after 10 minutes the tubes are taken out and the solutions are read using a colorimeter. Now you can see the purple color developed in the tubes. I am showing here blank, S1, S5 and one of the tests also. Now we are doing the blanking of colorimeter. 1 ml of blank is added to a clean cuvette. 1 ml is the reading volume of colorimeters. Now this is wiped, the cuvette is wiped clean of any moisture, that is important, and inserted into the chamber, reading chamber of colorimeter. The machine is set at wavelength of 540 nanometers for doing BIURET tests. Calibration is done by pressing 0 button, and now 0 calibration is done. Now we read all the solutions one by one, and any remnants of the previous solution is removed by tapping on the tissue paper. And 1 ml each of this S1 to S5, T1, T2 are read using the same procedure. We need not press any buttons on the colorimeter. The reading will automatically appear in the display. We can see that. This is the tabular column. You have to pause the picture and learn it well, study it well. All the rows and columns are given, the volumes added are given, and the final row we can see the optical densities obtained. In the second row there is a calculation, the amount of standard added to S1 to S5, from S1 to S5. That calculation is also given in the explanation, so that also should be learned well. And I'm using the values of S5 and T1 to calculate the final result, and the final result got was 1.67 milligrams of protein per ml of the solution, which means 167 milligrams of protein per 100 ml. Now finding the result from graph, on the x-axis concentrations, on the y-axis optical densities, spots are made and curve is made, and from the absorbance or optical density of T1, yes, an absorbance of 0.27 gives a result of 1.67 milligrams per ml. Now, the principle of BIURET reaction. BIURET reagent actually contains copper sulfate, sodium potassium tartrate, potassium iodide in 0.1 normal NaOH. the CO2 plus divalent copper cation, which is present in BIURET reagent, react with peptide bond nitrogen in alkaline medium to give a purple blue colored complex. The color can be read at 540 nanometers using a colorimeter. Now, a single copper ion C2 plus react with as many as 6 peptide bond nitrogens. So, if more number of peptide bonds are there in the given solution, the higher will be the intensity of the color of the final solution. And this also should be noted that peptides having at least 2 peptide bonds only will react with BIURET reagent to give a positive BIURET reaction. So, amino acids or dipeptides won't give you a positive BIURET reaction, but tripeptides give positive BIURET reaction. Now, in our experiment, the second unknown reagent, that is unknown test, that is T2, was having an absorbance of 0.22. You can calculate the result yourselves. So this is a quantitative estimation of a protein using BIURET reaction. Thank you for watching my video.